Hi, everyone. Welcome. Today we have as our speaker Eiman Azim. He's an associate professor and the Scanling Chair of Neurobiology at the Salk Institute. He earned his bachelor's in biology and philosophy of science from Stanford. While he was an undergrad, he discovered that human females respond differently than males to humor. Females exhibit a greater degree of language processing and executive control. Next, he went to Harvard and did his PhD with Jeffrey Mackless, studying how excitatory and inhibitory neurons differentiate during development. Finally, he completed postdoctoral training at Columbia University with Tom Jessel, studying the motor information in the spinal cord and how it's sent back to the brain via propriospinal neurons and the role that is played in online control of movements. His lab at Salk now explores how the nervous system controls dexterous movements, taking advantage of mouse genetics, electrophysiology, imaging, behavioral assays, and computational modeling. Excited to have you with us, Simon. Thank you so much for your time.
Great. Thank you very much, Reza, and thank you everybody for coming. It's really an honor to have the opportunity to present at this fantastic series of talks on this very normal and inconsequential Tuesday. So thank you for having me. So my lab here at the Salk Institute is interested in how the brain and the spinal cord control the body. And in particular, we focus on dexterous movements of the arms, the hands, the fingers. So these types of behaviors, they've fascinated artists and philosophers and scientists for millennia, and for good reason. In many ways, they're the pinnacle of what our bodies can achieve. And if you think about even these everyday tasks, like typing a text on your phone or buttoning your shirt, these movements are quite remarkable. Somehow we're able to coordinate dozens of muscles with exquisite speed and precision to achieve what we want to achieve. And so the overarching theme of what we work on in my lab is to try to figure out how we establish this fidelity between what we want to accomplish and what our bodies can actually do. So allow me to dramatically overly simplify things, especially for this audience, and start to sort of build up a little bit of a schematic and outline inspired by some ideas from Reza and from many others from control theory about how the motor system might work to move your limbs. So of course, to move your limbs, we think that supraspinal centers in the brain, the cortex, the brainstem, cerebellum, many other regions send descending pathways into the spinal cord, by which they convey motor commands to spinal circuits. Ultimately, the only way that you can get the muscles in your limb to contract to recruit the final common pathway, the motor neurons, of course. But equally important to this constant sort of feed-forward output of command is the constant influx of feedback from the periphery, feedback that comes back into the spinal cord to recruit reflex pathways, to regulate central pattern generating circuits. But of course, much of this feedback also ascends up into the brain, where from a control theory perspective, we think that this is helping to inform our estimate of the state of our body. And if we can constantly update the estimate of the state of our body, then in principle, we can update these ongoing motor commands and sort of in this sort of virtuous loop between output and feedback. Now, I think if things were this straightforward, we as a community would have largely figured out motor control by now, but of course things are much more complicated. So I want to point out a couple challenges the sensorimotor system has to solve that my lab has decided to try to tackle. So the first is that this idea that we're inundated with sensory feedback. If you think about every single receptor on your skin, for example, all of the information that's conveyed from those afferents can't gain access to all the circuits in the central nervous system. Most of that feedback is distracting or disruptive, so we need mechanisms to attenuate that distracting feedback while facilitating the salient information. Now, even if we have appropriate levels of gain control, even if we can adjust the amount of feedback coming into the central nervous system.
The one I sent you, Eiman Azim.
Hello? Okay, so even if we can adjust the feedback that's coming into the central nervous system, another major challenge that the sensorimotor system has to solve is that this feedback is, of course, inherently delayed. It just takes time for feedback to get from the periphery back into the central nervous system. And as many of you know, these delays, again, from a control theory perspective, introduce a lot of problems. But just said sort of more intuitively, if I'm sitting here trying to reach for my coffee cup, this reaching movement takes about 150 to 200 milliseconds. And we can achieve pretty exquisite endpoint precision in these behaviors. But it's hard to imagine how we can use sensory feedback that's delayed dozens of milliseconds, if not more, to be updating our state estimates to achieve such fine online adjustments. So an idea that's been around in the field for a long time is that we can compensate for this delayed sensory feedback with a faster form of information that takes the form of efference copies or internal copies. So the idea here is that as the commands are conveyed down to the spinal cord, and before they get to the motor neurons, they branch off at different levels, perhaps every single level of the sensorimotor system, and send copies of these commands to the cerebellum. And the cerebellum is thought to have learned an internal model of the physics of the body and how it interacts with the world across your lifespan. And this is a very malleable model, of course, as we all know. But the idea here is that if you can send a copy of your ongoing motor command to the cerebellum, you can implement what's called a forward model and essentially generate a prediction of the impending movement outcome. So if you can predict what the outcome of the subsequent movement is going to be, then you can update your state estimates much faster by compensating for this delayed feedback, and therefore update your motor commands more quickly. Now, of course, our predictions aren't always perfect. We'll often miss the free throw or even miss our coffee cup, but it's better to have predictions than to not have any predictions at all and only rely on delayed sensory feedback. And so on very rapid timescales, we could, in principle, use these predictions for rapid online refinement, which is going to be most of the focus of the talk today. On longer timescales, as many of you study, we can also compare these predictions to the ground truth, the actual movement outcome that's delivered by this delayed sensory feedback. And mismatches between prediction and outcome can be corrected for subsequent movements through a process called sensorimotor adaptation. Okay, so we're really compelled by these ideas. Many of them remain largely theoretical, but have a lot of experimental evidence thanks to many labs here and many labs out in the community. But the way that we try to tackle this, try to find the implementation level, the hardware that might establish mechanisms for gain control and efference copies, is to, of course, use mouse genetic tools where we can leverage the fact that different types of neurons express different genes. We piggyback on that molecular heterogeneity to try to gain access to specific neural circuits. We use that for perturbations, of course, but we also combine this with a variety of high-resolution 3D behavioral assays in the lab where we ask animals to perform these dexterous behaviors. We do 3D kinematic tracking. We'll record for many muscles for EMG recordings and also record in the central nervous system, all to try to combine all of this data to try to get a better sense of how neural activity establishes motor output. A branch of my lab is occupied with sort of developing tools to try to make the ways with which we gather behavioral data and analyze behavioral data a bit better. So I'm not going to talk about this today, but here's a paper we published last year where we propose a method for dramatically improving the ways that you gather training data for training these pretty revolutionary machine learning based approaches for kinematic tracking. Okay, so what I want to talk about today is first is very briefly just touch on some of the aspects of gain control that we've been investigating in the lab, and then spend the majority of my talk more really sort of focused on cerebellum and looking at one project that's looking at the organization of internal copy pathways as they come into the cerebellum, and then sort of ending the talk with a story where we're looking at how the output from the cerebellum, from the cerebellar nuclei might be organized for this rapid online refinement. Okay, so I first got interested in gain control when I was a postdoc at Columbia in the Jessel lab, where we looked at how you might have neural mechanisms to adjust sensory feedback gains at what's perhaps the best understood and simplest circuit in the nervous system, the monosynaptic stretch reflex. And so this is where proprioceptors come from the muscles and come into the central nervous system and directly innervate the same motor neurons that go back to innervate that same muscle. And so I'm not going to show you any data. This was published quite a while ago, but long story short, we found a way to genetically identify and access a specific class of inhibitory neurons, presynaptic inhibitory neurons that can regulate sensory gains in the spinal cord. When we eliminated those neurons, mice developed these very severe oscillations, these 20 hertz tremors. And so basically what we concluded is one of the major functions of gain control at the lowest level of the nervous system, at the stretch reflex pathway, is to establish limb stability. But of course, if I want to type an email or grab my coffee cup, I need more than limb stability. We need mechanisms to distinguish the distracting from the salient. If I'm playing the guitar, for example, I'm not paying attention to the weight of the guitar on my leg or the feel of my collar on my neck, but I need to pay exquisite attention to the salient feedback from my fingertips. We also have to distinguish the predicted from the unexpected. So in our field, of course, this is reafference versus exafference. We generate a huge amount of feedback when we move that from the sensory afference perspective is indistinguishable from signals that are generated by the external environment. So our central nervous system needs mechanisms to distinguish this reafference from this exafference and in general attenuate the reafference. And so in my lab, we got interested in, well, what about all of this feedback that's coming up and entering the brain? And we decided to look at the very first synapse that touch feedback from the hands, tactile feedback, makes in the central nervous system. And so just a bit of a refresher from textbook anatomy. This is the medial lemniscal pathway. So the sensory afferents that convey touch information sit out in the periphery. Their cell bodies live in the dorsal root ganglia. They send their axons directly into the spinal cord, but also send axons that project directly up into where they make their first synapse into the brainstem in the dorsal column nuclei. And the cuneate nucleus in particular is tasked with receiving feedback from the hands, the arms, and the upper body. Here in the cuneate, these afferents form synapses onto cuneo-lemniscal neurons that convey this information to the contralateral thalamus, which then sends this information to the primary somatosensory cortex. So Jim Conner and lab decided to tackle this question and ask, are there mechanisms for regulating sensory feedback gain at this first synapse? So we published this a couple of years ago, and I'm not going to go into any of the data. I just want to summarize what we found because it kind of leads into some preliminary data we have now that's more relevant for cerebellar circuitry before jumping into some of the questions about efference copies. So in this project, the first thing that Jim asked is, are there mechanisms for regulating the strength of tactile feedback at this very first synapse in the brain, at the cuneate nucleus? And indeed, we found a heterogeneous group of inhibitory neurons, both GABAergic and glycinergic, that sort of surround the core of the cuneate, surround these cuneo-lemniscal neurons, and can regulate the amount of how responsive these cuneo-lemniscal neurons are to a given tactile input. Then through in vivo studies, we found that indeed, if you modulate these inhibitory neurons, you can regulate bidirectionally the amount of feedback that's transmitted through the cuneate up to the thalamus. And if you do this and push the activity of this inhibitory shell in either direction, either too much activity or too little activity, you have pretty severe consequences for behaviors that require tactile feedback from the hands. I'm not going to go into any detail, but I'm happy to address any of this in the Q&A if you'd like. And finally, what we found is that there are at least two major cortical top-down circuits that directly innervate these cuneate circuits. One from the primary somatosensory cortex that directly innervates the cuneo-lemniscal neurons and can directly excite these channels that are providing tactile input to the cortex. And another much larger population of neurons in the more rostral cortex that projects down, completely avoids the cuneo-lemniscal neurons and directly innervates these inhibitory neurons and can provide feed-forward inhibition onto this cuneo-lemniscal neuron. So long story short is we think we found these two opposing top-down pathways that might be able to sort of exert a push-pull on attenuation and facilitation of incoming tactile feedback. So in ongoing work in the lab, we're now recording from awake behaving animals as they perform tasks where they're generating reafference or where they need to be sensitive to exafference, touch feedback on the hands, with the hypothesis that this large rostral population that can elicit feed-forward inhibition might be relevant and might be active during movement and even during the planning phases of movement to attenuate the huge amount of reafference that occurs during movement. So we're pursuing those ideas now. So a little bit of just sort of unpublished data that as we follow up on this work, we've also identified that this doesn't apply only to the hands, perhaps unsurprisingly. We can find a similar inhibitory circuitry that can attenuate feedback from all over the body. In fact, if anything, on hairy skin, it's even more potent, which may not be surprising when a mouse is moving through the world and all of the hairs are moving. They generate a huge amount of cutaneous feedback, and so there are mechanisms to attenuate how much of that feedback can make it through the dorsal column nuclei. If you move more laterally to the trigeminal system, we can find inhibitory circuits that can attenuate feedback from the vibrissa. And so there's sort of this entire map of attenuating circuits, inhibitory circuits in the brainstem that, again, at this very first synapse in the nervous system, can serve to regulate sensory feedback. And of relevance, I think, to this community in the cerebellar world, we've also been looking at the neighboring external cuneate, which has a very similar name, but as many of you know, is a very different structure. The external cuneate receives direct input from the proprioceptive afferents of the upper body and the forelimb. And these proprioceptive afferents form synapses in the external cuneate, which is composed of mossy fiber projections to the cerebellum. Here you can see the projections of external cuneate neurons into the cerebellar cortex. And essentially by doing an assay where we're recording in the external cuneate while generating proprioceptive responses, we can also find a sort of inhibitory neurons that are local that can also elicit the same kind of attenuation of proprioceptive feedback. So we think what we're identifying here in the brainstem, again, at this first stop of this sensory information, is a more general circuit motif for somatosensory modulation more generally. And what we're really interested in doing is trying to identify the different top-down inputs that can sort of pull those levers of modulation to attenuate the disruptive and facilitate the salient. Okay, so for the rest of the talk, I want to switch gears and really focus on some of our more cerebellar focused questions. And so it's coming back to this idea of internal copies. So again, a reminder, the idea here is that we send copies to the cerebellum to generate online predictions of movement outcome that can be used for rapid online refinement of movements. So I'm going to talk a bit about a project that we're working on looking at a putative pathway from the spinal cord through the brainstem into the cerebellum that we think is conveying this copy information. And then I'm going to flip to the other side of the cerebellum and ask, how does cerebellar output from the cerebellar nuclei elicit really rapid online refinement? Okay, so looking at these copy pathways, we've decided to focus on a structure in the very back of the medulla called the lateral reticular nucleus. And the reason we look at this structure in the brainstem has to do with decades of really beautiful work from a collaborative mind, Bror Alstermark, and his mentor, Anders Lundberg, who identified this class of neurons in the spinal cord that they termed C3, C4 propriospinal neurons. So this is a subset of a much bigger class of propriospinal neurons. And what makes these neurons unique is they sit in the cervical spinal cord. They get convergent descending input from command pathways from the cortex, the brainstem, et cetera. And then what makes these neurons really interesting is they have an axon that bifurcates. So from a single propriospinal neuron, the axon comes out, bifurcates. One branch projects directly to the motor neurons that innervate the forelimb muscles. And the other branch ascends up into the brainstem to the LRN, the lateral reticular nucleus, which is made up essentially entirely of mossy fiber projections to the cerebellum. So anatomically speaking, it looks like these PNs are well sort of anatomically set up to convey commands down to motor neurons and copies of these commands to the LRN. This is in another project when I was a postdoc in the Jessel lab, we identified a subset, an excitatory subset of these propriospinal neurons within the V2A class in the cervical spinal cord. I'm not going to show you any data except one electrophysiological experiment. So what we found is that one, if we ablate these propriospinal neurons, mice become very dysmetric. So they really need these neurons to perform accurate reaching movements. But then if we go into anesthetized animals and we selectively activate just the ascending branch that goes to the LRN while recording intracellularly from motor neurons in the cervical spinal cord, what we find is that we can elicit EPSPs. We can modulate motor neuron activity extremely rapidly within about 5 milliseconds. And so what this told us is that these propriospinal neurons are well placed to convey information that can come up into cerebellar circuits, very likely through the cerebellar nuclei directly, and come back down extremely rapidly. So if a mouse, just like a human, is performing a reaching movement in about 150, 200 milliseconds, a pathway that can convey copy information up and then regulate motor neuron activity very quickly, every 5 to 10 milliseconds or so, would be very useful for rapid online refinement. So what I'll tell you a bit about is our work trying to dig into the LRN. We've been recording in the LRN to see if there's any logic to its activity during these reaching behaviors. We've been perturbing the LRN to see what contributions it might have to limb movement. And finally, in a separate sort of side project, we've been looking to see if there's any molecular and functional organization within the LRN, any heterogeneity. So in thinking about the LRN, my collaborator, Bror Alstermark, wrote a really nice review towards the end of his life with Carl Friedrich Eckeroth, basically outlining how the LRN might be organized. So one idea is that the LRN conveys information, this copy information, in sort of a label line fashion. So you don't really need to pay attention to all these acronyms. You can think of these inputs from the spinal cord as conveying information from different pathways, for example, from different motor pools or from different segments of the spinal cord. And one idea is that this information is conveyed in a label line through the LRN up to the cerebellum. And one of the very few papers out there that's recorded from the LRN, this is a fictive locomotor prep in a cat. So it's a decerebrate cat where they've evoked fictive locomotion by stimulating the MLR. What they found is some evidence that LRN neurons prefer to respond their activity correlates better with either extensor or flexor activity. And so if you record from enough LRN neurons, you can see their activity sort of tiling the space of these fictive locomotor movements. So this provided some evidence then maybe there is sort of a label line with some LRN neurons preferring flexor and some preferring extensor information. And so if we were to record in awake behaving animals during a reaching movement, for example, what we might expect to see are neurons that fire at different phases of the movement, again, sort of tiling the space of the behavior. Now, alternatively, you could have some integration of this information, some computation happening in the LRN. Very few things, if any things, I think can legitimately be called a relay in the nervous system. If the structure is there, it's probably there for a reason. So one possibility is that there is some sort of integration of information from these different spinal segments or different motor pools coming into the LRN, and this integrated information is conveyed to the cerebellum. And if we were to record in the LRN in this scenario, we might see a more complicated pattern of activity that represents what might be thought of as sort of mixed selectivity with the same neuron firing at different phases of the movement. So we decided to try to record from the LRN, thinking, okay, no problem, let's go in and record. I think there's a reason that, to my knowledge, nobody had recorded from the LRN of an awake behaving adult animal because it's very hard to access. It's in the very back of the brainstem, essentially right where it's entering into the spinal cord. So it's very mobile. It moves a lot. You do a lot of damage if you don't record with the right tools. And it's also right next to breathing circuits. And so if you're not careful, of course, you're going to have some pretty deleterious consequences for the mouse. And so we had to sort of troubleshoot a lot of different ways to record. But Elijah, a former graduate student in the lab, Kiwi, a current postdoc in the lab, figured out a nice way to record and get a lot of sort of stable units as we're having mice perform these reaching movements. So essentially what we use is these sort of custom-made optetro devices. So we 3D print all these parts. It's essentially an optical fiber with 8 tetrode bundles affixed to the end of the fiber that protrude below the optical fiber. So there's a few advantages here. One, the optical fiber gives a lot of stability, so we can hold our units despite the mobility of the brain. The fiber can also be used to deliver light, so we can use light and optogenetics to tag neurons of interest, and we have genetic markers for LRN neurons. And finally, this whole device is on a microdrive. So even though from the tetrode bundles, you don't get the same kind of yield you'd get from a single neuropixel recording, we can move the drive down, 50, 60 micron per day. And from a single animal over the course of a couple of weeks, we can collect hundreds of different units because we can collect new units on every day of behavior. So we implant these optetrodes in mice performing head-fixed water-reaching movements in this case. We collect a lot of kinematic data. So we sort of have this pipeline now where we collect dozens of kinematic features, and that'll be relevant in a moment. And then what you can see is perhaps unexpectedly, or perhaps expectedly, is you see LRN neurons that correlate with different features of the behavior. We see neurons whose activity looks like it is modulated by aspects of kinematics like distance from the target or the speed of the limb movement. And if we look across all the units of, let's just take data from one mouse, we can find many units whose activity is significantly up or down regulated. If we align it to different events of the behavior, for example, the tone onset, the reaching movement itself, or the reward, contacting the water and retrieving the water, what we see is the activity best aligns to the movement itself. So a lot of these neurons really like to be active during the execution of the behavior, and their activity correlates with kinematics. But I think more interestingly, what we see is no evidence of this sort of tiling of different LRN neurons are active during different phases of the movement. We see a lot of neurons whose activity is up or down regulated throughout the behavior, suggesting this sort of mixed selectivity. So we're in the middle now of building generalized linear models with our sort of dozens of kinematic features. And what we're finding, as I just mentioned, is that there are neurons whose activity can be best explained by combinations of kinematic features of the movement. And I think most interestingly, in a lot of those neurons, the activity either is significantly up or down regulated in advance of the execution of that aspect of kinematics, suggesting that these neurons are conveying information about what the limb is about to do, which is exactly what you'd want an efference copy pathway to do. Okay, so moving to perturbation. Now we've been perturbing the LRN to see if there's any consequence for these limb movements. So again, we have genetic access. In this experiment, we've gone in and we've completely ablated the neurons using a selective diphtheria toxin approach. We have the mice perform these reaching behaviors. And I think what surprised me most is that the phenotype was very minimal. The kinematics looked relatively normal. If anything, we saw a bit more variability in endpoint precision in these behaviors, but it was quite subtle, which surprised us because there's a handful of classical studies in cats using things like electrolytic lesions to go in and ablate the LRN, and the phenotypes were very severe. But we suspect that that those lesions may not have been quite specific enough. And when we use the genetic specificity we get here, we actually see pretty minor phenotypes. And I'll get back to that in a moment. So this took us on a bit of a sort of tangent. We decided we were kind of frustrated with looking at all of our behavioral data sets, and these data sets are constantly growing because we collect more and more of these kinematic features. And it became harder and harder, as you can expect, to decide by eye what the phenotype is. So we wanted to develop an approach that is a bit less biased and a bit more comprehensive. So instead of looking under the lamppost, we have sort of a floodlight that we can shine around to help us focus where we should be looking for phenotypes. So what we do is we essentially take all of those kinematic features I mentioned. So again, we have dozens of features about position, velocity, acceleration, about all of these different landmarks on the limb, joint angles, and other data sets. We have EMG data. You can take any sort of time series data, and nothing new here. What we do is we break it into two groups and we train a classifier, in this case, a random forest classifier with training data. And then we apply test data, held out data to the classifier. Again, that's not a new approach. And what you can essentially do is ask the classifier if on the held out data, it can detect a difference between, for example, pre and post ablation mice. And the higher this AUC score is, the better the classifier is at detecting a difference, the more likely there's a phenotype there. Great. But what we really cared about is if there is a phenotype there, if there is a high AUC score, what's explaining the phenotype? So to try to get at that, in parallel, we use an approach called SHAP, or Shapley Additive Explanations, where we essentially train this sort of explanatory model with the exact same kinematic features. And what we can do here is essentially generate a rank-ordered list of which features contribute most to that AUC score. So these features at the top can best explain the phenotypes we're seeing. So an example of what we can find is here, we're taking 8 mice that are ablated, have LRN ablation, 8 mice that are control. The thick lines are the means. The AUC scores are high for all the red mice, suggesting that there is a phenotype there. And then what we can do is we can take the entire reach, or in this case, we can take just the late phase of the reach, for example. So just the last, I think this is the last 20% of the trials of the reaching movements, apply our SHAP model. And what we can generate is a list. I'm just showing just a handful of the kinematic features here with the features that contribute most to that AUC score shown here with the largest bars. And so in these mice, it looks like, as suggested by that initial data I showed you, that endpoint precision is most affected in these animals. And so it was at this point that now we sort of have this way to do a more comprehensive description, but we realize that the phenotype, if it's there, is very subtle after ablating these neurons. And we got concerned that what we do in those ablation experiments is we kill the neurons. We give the animals, about a week before testing them again. And as we all know, that the cerebellum is very good at functioning like an adaptive filter. It's very good at dealing with aberrant inputs over time and adjusting its output to accommodate. And so we worried that by giving the animal a week, it's compensating in one way or another, because we're not eliminating all mossy fiber inputs or all efference copy inputs, just the ones from the LRN. So what we decided to do is take a much more acute approach. So here we're going to use optogenetic silencing to essentially put an inhibitory opsin in the LRN neurons. Here, this is just recording the LRN, showing that our silencing is very effective. Now, here's an animal with the light off, performing a normal reaching movement. This is about, I think, 15% real time. And we turn the light on. Same mouse, videos at the same speed. I think you can tell that the most obvious phenotype here is they move much more slowly. The duration of the movement's much longer, but they also have deviations in their trajectory. So this sort of combination of worse trajectories and slower speed leads to much longer duration reaches. And we apply our SHAP analysis, and indeed what we see are a lot of aspects of velocity that come out as explaining the phenotypes. And so our interpretation here is what we're doing is we're removing a major, but not the only major efference copy input to the cerebellum. And what the sensorimotor system is left to do is to use what remaining information it has, which is almost by definition going to be longer latency pathways. These could be efference copy pathways, for example, through the pontine nucleus that are going to, that input's coming from the cortex and eventually probably going to go back through loops up to the cortex. So this is going to have a longer latency to elicit impacts on motor output, or of course, sensory feedback, which of course is also slower. So we think what's happening is that the sensorimotor system is now resorting to the information that it has that's now slower, and that's causing the animal to move more slowly to achieve precision. And this is not an entirely new phenomenon. If you look at patients with cerebellar ataxia, for example, in conditions where they really need to achieve endpoint precision, they often will move more slowly to achieve that precision presumably because their predictive control is somewhat impaired. Okay, so in a sort of separate project, we've also been trying to identify whether we can find any molecular or subtype organization within the LRN. So classical anatomical studies in many species have suggested that the LRN can be broken into different parts, a magnocellular or a parvocellular part, for example, that tends to get inputs from the cervical or the lumbar cord. So we decided to use some sort of modern molecular approaches to sort of ask these questions. So we isolate LRN neurons, we do RNA sequencing. I'm not going to go into any of the details. I think it's probably familiar to many of you. It's a very common approach these days. We do dimensionality reduction to identify whether any clusters there. And in this case, Kiwi, who's led this work, identified two genes, PCP4 and HTR2C, that seem to distinguish the magnocellular and the parvocellular part. So the magnocellular, again, we think is getting input from cervical, HTR2C is expressed here in the parvocellular, and they're essentially non-overlapping. Using intersectional viral and genetic approaches, we can selectively gain access to these magnocellular, or in this case, the parvocellular populations, and start to ask questions about function, which we're in the middle of doing. But as a first pass, what we decided to do is just look if there are any anatomical distinction. So we use a monosynaptic rabies tracing approach, which was developed here at the Salk by Ed Callaway, to essentially ask which neurons provide direct input to either all of the LRN neurons here, or just that parvocellular population, that HTR2C population. We can already detect differences in the brain. We see a lot less cortical input, we think, to the parvocellular population. And if we look in the spinal cord, what we can see is that if you look at all the LRN neurons, as expected, as has been reported in the literature, most of the inputs come from the cervical spinal cord. But if you label the inputs coming just into that parvocellular HTR2C population, what you see is a very disproportionate amount coming from the lumbar cord, suggesting that these neurons really prefer to receive information from the lumbar cord and might convey hind limb-related efference copy information to the cerebellum. So we're in the middle of sort of digging in to any functional differences between these two populations right now. And so finally, I want to tell you about a story that we're wrapping up now, looking at the other end of the cerebellum. So if the cerebellum could, in principle, take these copies, generate predictions of movement outcome, the cerebellum needs a way to rapidly refine ongoing motor output. So how can it do that? Well, of course, the only output from the cerebellum that can influence limb movement is from the cerebellar nuclei. In mice, these nuclei are called the medial, which of course in primates is vestigial, the interposed, and the lateral, or the dentate in primates. And as we all know, these output structures have very diverse targets. This is just a non-exhaustive list from a review that Aisha Thanawala, a postdoc in my lab, put together a few years ago. And so you see targets in the thalamus, the midbrain, the brainstem, and the spinal cord, as I'll get to in a moment. If you look at how people have studied limb movements with respect to cerebellar nuclei, much of the work has been done looking at the interposed. So there are classical and really sort of foundational studies, for example, in this cat study doing mucimolin activation in the interposed nucleus, showing that when the interposed is inactivated, these cats become very dysmetric and they would overreach or underreach the target. Using mouse genetic tools, Aisha, who I just mentioned, is a postdoc in my lab, when she was a graduate student in Albert Chen's lab, she found a genetically distinct class of interneurons of anterior interposed neurons that project to the thalamus. She was able to ablate these neurons, and when this specific subset of anterior interposed neurons was ablated, the mice became hypermetric. They would overreach the target, and when she activated these neurons, it became hypometric, or they would underreach the target. Soon after, Abby Pearson had a really nice paper, again, going into the anterior interposed. And in this case, what she was doing, Matt Becker, her student at the time, was activating these anterior interposed neurons using optogenetic approaches and showing that when these neurons were activated, there was a deceleration of the limb, which of course, in the end, will cause a hypometria. So together, these studies suggested there's a major class of neurons in anterior interposed that might be tasked with decelerating the limb, perhaps to achieve endpoint precision. So we went into this project thinking, okay, well, both of those projects were looking at and focusing, and in fact, much of the field is focused on projections from the cerebellar nuclei to the thalamus. So maybe these longer latency pathways that go up to the thalamus, cortex, and eventually back down are involved with the really sort of sophisticated things like endpoint precision that require a lot of coordination across segments of the limb. And so we decided to compare two extremes. So if you've got these thalamus projecting neurons. What about all these neurons that project directly to the spinal cord? These have been far less well described. There was really only one paper studying these neurons in mice from Ariel Levine's lab a few years ago. And what she found is that there are neurons in the cerebellar nuclei that project to spinal cord. And when she used a chemogenetic approach, these dread receptors, to hyperpolarize the neurons, reduce their activity, she found deficits in reaching behavior, suggesting these neurons might be important for controlling movement. So we wanted to come in and use electrophysiological and optogenetic approaches with sort of really high temporal resolution to ask questions with the hypothesis that if these are important for longer latency endpoint precision, maybe these neurons are really important for the constant, really rapid online adjustments throughout the course of the reaching behavior. Now, our hypothesis ended up being wrong, and I'll get to that in a moment. But before I show you some of that data, we first wanted to get sort of a comprehensive perspective of what we're looking at. So Oren, a grad student in the lab, essentially generated a quantitative map of all of the excitatory output in this VGLUT2 create mouse, of all the excitatory output from the cerebellar nuclei. So here are injections in the nuclei themselves, and you find all the usual suspects. You see thalamus, motor thalamus, red nucleus, targets in the brainstem, and as I just mentioned, targets in the spinal cord. I want to zoom into these targets in the spinal cord a bit more because these have been far less well described. So the first thing we did is we went in and we recorded in the spinal cord from interneurons extracellularly. And what we found is that while electrically stimulating in the cerebellar nuclei, Johan Zhang, our collaborator who trained with Bor Ostermark in Sweden, identified interneurons that get direct monosynaptic input. So indeed, these cerebellar nuclei neurons project down to the spinal cord and directly innervate and excite interneurons in the spinal cord. Incidentally, we found no direct inputs to motor neurons. All the effects were at least disynaptic. We also did some rabies tracing showing that interneurons in the spinal cord, either excitatory or inhibitory, or some of our favorite subclasses like the V2As and the V1s, also get direct input from the cerebellar nuclei, especially from the posterior interpose, which is going to be important in a moment. Okay, so back to our hypothesis. Well, maybe these thalamic projecting neurons are important for endpoint precision, and maybe these are important for rapid online control. But things got more complicated pretty quickly. So Aisha was doing some dual retrograde labeling experiments. So putting one label in the cervical spinal cord, another in motor thalamus. And what she found is a lot of neurons in green that go to the thalamus, a lot of neurons in red that go to the spinal cord, but also quite a few neurons in yellow, especially in posterior interpose and in the medial nucleus, that project to both targets. So you've got a lot of these neurons that send axons that are at least bifurcating and going both to thalamus and spinal cord. And so we quickly realized, well, we won't be able to ask anything about these neurons if we just use this sort of unilateral approach of just targeting spinal cord or thalamus. We need to use a combinatorial approach to get selective access to different classes of neurons with more specificity. So to do that, we've been using this intersect viral approach developed in the Diceroth lab, where essentially you can apply this Boolean logic to your anatomical targeting. So I'll show you, for example, we can target neurons that go by definition to thalamus, but do not go to spinal cord. And so here you can see the neurons in the cerebellar nuclei are targeted. They go to motor thalamus, red nucleus, but we've deleted the neurons. We do not access the neurons in this experiment by definition that go to the spinal cord. We can also do a sort of AND experiment. What about neurons that go to both, these dual projecting neurons? And again, here, what we can see are neurons that go to a more restricted region of motor thalamus, a more restricted region of the red nucleus, different parts of the brainstem, interestingly, and also go to the spinal cord. And then we can also apply this to the spinal projecting neurons and remove all the ones that go to thalamus. So we can take out all the ones that go to thalamus and just look at the neurons that go to the spinal cord. Now, doing all this anatomy, we realize that we need to make quantitative maps. I'm a fundamental believer in the idea that anatomy is really foundational if we want to understand at an implementation level the function of these circuits. So Eliza Rhee, a really talented research assistant in the lab, has been making a really full quantitative map across the brain and the spinal cord. I don't expect you to read or interpret all these acronyms from the Allen Institute's atlas, but this is just to say that these neurons, which you'll see here called the CNVAL, these are the ones that go to thalamus and not spinal cord. And these neurons, which I'll just call the duals, but these are the ones that go to the thalamus and the spinal cord, have dozens of collaterals on the way down. I'm just showing you some of those collaterals, both on the contralateral and the ipsilateral side of the brain. And so through all these quantitative maps, we've been able to generate sort of these maps of these different populations of neurons and all of the targets that they innervate. So here are those neurons that go to the thalamus and don't go to the spinal cord, but they send collaterals to the red nucleus, to the brainstem, and many other targets. You can see these dual neurons. They go to thalamus and spinal cord. They also go to red nucleus, but they also innervate different targets in the brainstem, which we think are really interesting. And then you see these ones that only go to the spinal cord, not the thalamus, and they don't really innervate anything else in the brain that we can see. And so the point I want to make here is that the anatomy is daunting, and we're going to have to grapple with the fact that our sort of textbook ideas that, you know, dentate goes to thalamus, interpose goes maybe red nucleus, medial goes a lot to the spinal cord are too simplistic if we want to understand the function of these neurons. They have huge numbers of collaterals. What we're showing here are quantitatively, the thickness indicates the proportion of the collaterals that go to each of these targets. And the colored lines indicate things that are unique, that are not shared by the other populations. So there's a huge amount of anatomical diversity. And importantly, the only way you'll be able to distinguish these two is to use these combinatorial viral approaches I was saying, because both of these projection neurons go to the thalamus, but as you'll see in a moment, they're quite different functionally. Okay, so for the last few slides, I just want to focus just on these neurons that go to thalamus and not spinal cord, and these dual projecting neurons. So Aisha's been recording from mice performing these head-fixed water-reaching movements, while also doing EMG recording from many forelimb muscles. I'll just show you data from 3 of these muscles. So what she's done is she's introduced, using this combinatorial viral approach, channel rhodopsin to activate either these thalamus projecting neurons or these dual projecting neurons. And what you can see is that when she turns the light on, the kinematics are affected. So the light turns on every time there's one of these red plus signs. But I don't think you can really get much just by looking at these 3D or 2D plots. Let me just show you a couple movies. So that's a normal reaching movement. Now we're going to activate specifically these thalamus projecting neurons. You'll be able to tell when the light's on here at 18 hertz. You can see every time we activate these neurons, we see this retraction of the limb, this flexion of the elbow as the limb moves backwards. And now if we activate these neurons, these dual neurons that go to thalamus and spinal cord, you see essentially an opposite effect. The limb extends outwards. And interestingly, you also see this coordinated extension of the digits as the animal is reaching towards the target. If we look in more detail at the kinematics, we can see, as you'd expect, when we activate, we generate more sort of sub-movements, these corrective sub-movement. After the first stimulation, what we see is that the velocity, the change in the forward velocity is reduced when we activate these neurons. Again, indicative that the limb is sort of pulling backwards. The outward velocity is reduced, and the limb spends more time in this sort of posterior position closer to the body. And the exact opposite is true with these dual neurons. There are more sub-movements again, but when we activate after the first stimulation, there's an increase in outward velocity, and the limb spends more time away from the body. And so, as I mentioned, Aisha's been doing a lot of EMG recording from several muscles in the forelimb. What we find when we activate these thalamus projecting neurons is that when the light is off, of course, you see biceps and triceps activity. The animal is moving towards the target. But when we're activating, we see this sort of time-locked activation, specifically in the biceps and not the triceps. And again, the biceps is going to flex the elbow and pull the limb backwards. If we activate these dual projecting neurons, we don't really see much in the biceps beyond normal activity, but we see pretty clear time-locked activity in both the triceps, which is going to extend the elbow and push the limb outwards, as well as the anterior deltoid, which lifts the limb sort of upward and outward. And so what we think is happening here is that these neurons can recruit sort of coordinated motor pools across the limb to generate this outward movement. And in data I'm not showing, we also see that digit extensors are also activated, sort of pushing the digits outward as if the animal is anticipating and coordinating the outward reaching movement to grasp a target. Now, if we look at the EMG activity itself, at the latencies, what really surprised me is that we would see EMG activity extremely rapidly in about 5 to 10 milliseconds. And so what that means is these signals are getting from the cerebellar nuclei out to the muscle in 5 to 10 milliseconds. I didn't quite believe it at first, so we wanted to confirm. So what we did is we went in with Juan and we recorded from motor neurons themselves while stimulating the cerebellar nuclei. And what we see is, again, there's no monosynaptic inputs from the nuclei to the motor neurons, but we see these disynaptic responses in as little as about 3 milliseconds. So a signal can get from the cerebellar nuclei to motor neurons disynaptically in about 3 milliseconds, out to the muscles in a total of about 5 to 10 milliseconds, and then about another 15 milliseconds or so for that arriving action potential from the motor neurons to be translated into joint work. So extremely rapid. And again, we wouldn't be able to see these opposite effects if we didn't use a combinatorial approach to tease apart the sort of complicated anatomy of these output projections. So Akira, postdoc in the lab, has been building a biomechanical model of the limb using these hill-type muscle models with 3 pairs of extensors and flexors at the shoulder and elbow to see if we can sort of take our real EMG data, plug into our models, and recapitulate some of these activities and ask it some questions in a virtual environment. And finally, the last data I want to show you is from Oren, grad student in the lab, who's been recording using these optetro devices in the cerebellar nuclei. As many of you would expect, we see neurons in the cerebellar nuclei whose activity is modulated by movement. In this case, activity increases pretty significantly during reaching. We found a lot of neurons whose activity is significantly upregulated and a lot of neurons whose activity is significantly downregulated during the course of the reaching behavior. Oren's done some cross-correlation analysis between firing rate and outward hand velocity and can find positive and negative correlations. So neurons whose activity correlates with an increase in outward velocity, and conversely, neurons whose activity correlates with a decrease. And we find these sort of what we call anterior, posterior, positive or negative neurons, both in these activated and suppressed units during the behavior. And finally, if we dig in a little bit more, so I'll show you just an example unit. So here's an example unit. We've taken all of its firing across every single reaching trial that we've recorded. And then we break the firing rate into quartiles, where red is the highest quartile and blue is the lowest quartile. So red is when the neuron's firing the most. We normalize to the instantaneous firing velocity, firing rate. And what we ask is what happens to the limb velocity in the next 35 milliseconds. So when this neuron is in its highest firing rate, the limb velocity tends to increase subsequently. When the neuron is at its lowest firing rate, the limb velocity, outward velocity, tends to decrease over the next 35 milliseconds. We find many units with this profile. And if we look just at the highest quartile, so when this neuron is firing the most, where is the limb? So this is a sort of spatial density plot looking at where the hand is at that moment in time when the neuron is firing the most. And we see that it tends to be firing highest at the beginning of the reach, exactly when you're about to want outward acceleration towards the target. We also find many units that show the exact opposite. So here, highest firing rate corresponds to a decrease in outward velocity over the next 35 milliseconds. Lowest firing rate corresponds to an increase in velocity. You see the sort of mirror image here. So the highest firing rate has a decrease in outward velocity. And if we look at the spatial plot, we see that this neuron tends to fire the most when the hand is getting close to the target, exactly when you want some deceleration. So what we think we're finding, it's supporting this idea that these copies can be used for these rapid predictions and that the cerebellar output can very rapidly update motor output. But I think what's refining some of these ideas is that this output can completely bypass most of the brain. It can get back to the spinal cord extremely rapidly to do these biomechanically pretty sophisticated things that coordinate motor pools across segments of the limb to achieve endpoint precision. And so what we're really interested in now is seeing how these evolutionarily ancient pathways in the spinal cord, the brainstem, and the cerebellum can be used for really rapid adjustments through these loops that can be sort of complemented by these longer latency loops that most people have studied that go through thalamus and cortex to together coordinate these really dexterous behaviors. So to sum up, what I hope I've shown you today is that in my lab, we're combining anatomical and molecular, electrophysiological, behavioral, and modeling approaches to try to ask whether we can find evidence for or against these really influential control theory ideas like gain control and efference copies and forward models at the implementation level, at the hardware level. Again, focusing on the ancient evolutionarily conserved aspects of the motor system to try to get better insight into how all mammals might achieve this precision. But of course, eventually by comparing, contrasting, we hope to look at things that mice do not have that can help to explain how humans have used these evolutionarily ancient pathways and layered more sophisticated control on top of it to achieve this unparalleled dexterity. So with that, I want to thank everybody in the lab. I tried to highlight all of the really fantastic people who did the work that I presented today, and I'm happy to take any questions.
Oh, that was wonderful. That was amazing. If anybody has questions, just go ahead and please raise your hand and I will call on you. Let me get started, Eiman. We can go back to the LRN experiments, the optosilencing of it. I thought what was really interesting is that the limb slowed down drastically. And it made me think of Gordon Holmes and postural tone, thinking that one component of cerebellum's output may be into support the tone that you need in order to produce and maintain posture. And I'm wondering, when you stimulated during the movement rather than just from the beginning to the end, did you see a sort of like something consistent or perhaps against that idea of a postural tone?
That's a very good question. We're going exactly down that alley to try to have stimulation at distinct time points to see if there are different effects. To do that effectively, what we need to do is have these animals in our head fixed rig so we can very precisely time when the light comes on. For example, when it leaves the pedestal to start the reach, or we can do sort of closed loop silencing at different phases of the movement. So we don't have an answer yet, but I agree. I think beyond just looking at hand velocity and hand deviation, which we're doing now, our goal is to record for muscles and also look at aspects, for example, like you mentioned, of posture, because I think the LRN anatomically isn't just receiving information about the limb and about the hand, it's receiving information about from the entire spinal cord, and especially the cervical spinal cord.
Wonderful. Any questions for Eiman?
I have a question, but I don't have a hand. It's Sasha.
Hey, Sasha.
That was really beautiful, and there's a detailed question and a general question. So the detailed question is, did I see that the dentate or the lateral cerebellar nucleus in mouse projects to the spinal cord?
No, we see minimal.
Okay, maybe. Okay, I thought I saw that as great. Okay, good, because it should.
So
I mean, because
it's essentially
nobody else sees it. Right? Great, good. Okay, the bigger question has to do with why you would choose, other than technical advantage, the LRN, if you really want to know about fine control, because the LRN is doing this excitability tone thing, and inputs, those excitatory inputs directly to the cerebellar nuclei, which I love them, but they don't have the computational power that the cerebellar cortex does. There's just not room, right? So in my mind, and even from your work with Tom Jessel, it seems that there's sort of two sets of proprioceptors in a way. One that is about, and this relates to Reza brought up, one that's related to kind of tone, output tone, and the other that's the fine details. So you wouldn't expect from a priori, I don't think, the LRN to be doing the fine details. You might expect it to be doing the tone. And so somehow when I saw your results, which were beautiful, I wasn't at all surprised.
Yeah, no, that's a really good point. Let me make 2 points. One that I think supports what you're saying, and one that might contradict it a bit. So one, I think all of the really rapid, the really short latency effects I'm showing almost certainly are passing directly through the cerebellar nuclei and back down. At those latencies that I was showing, 5 to 8 milliseconds to get down to motor neurons, it's very unlikely that the cerebellar cortex is able to contribute. But that's not to say that it's not contributing soon after. So the LRN neurons, as you mentioned, send collaterals into the nuclei, but they also project all over the cerebellar cortex as mossy fibers targeting the granule cells. What I think is really compelling about the LRN is because it's getting so much input from interneurons in the spinal cord, it's in a place to get really granular, fine-grained detail about what the motor neurons are about to receive. I think the cerebellum, if it likes copy information, it wants copies at every granularity, and the propriospinal neurons in the spinal cord provide the finest granularity, the stuff the motor neuron is about to get. And so if you're thinking about dexterity, for example, motor pools that control individual digits, or wrist movements, for example, that's exactly the kind of information that spinal interneurons are well placed to convey to the LRN and eventually to the cerebellar cortex. Now, we haven't looked into the cerebellar cortex yet at what the LRN information is contributing, but I suspect beyond that tone function, by regulating the tonic cerebellar nuclei activity, that information is also being used for computation and by the cortex to regulate the nuclei activity. Does that make sense?
It does make sense. There is the little sticky point that all that, Eccles, this drove Eccles crazy on the output end. All of the fine granularity that you see on the inputs just gets lumped into some big, massive bilateral kind of output. And LRN's like that, right? Those neurons are huge, and so they're integrating a lot. So anyhow, it's beautiful work. Speculations are nothing compared to really insightful experiments. So keep going, fantastic.
Thank you, Sasha.
Thank you, Sasha. Any other questions for Eiman? Let me jump in. Eiman, you showed how ablation experiments and also optostimulation of the interposed, they were pretty consistent. They produced hypermetria of reaching. What do you think is, how do you put it together with what you were showing us with regard to thalamic, uniquely thalamic projections versus those that also go to the spinal cord?
Yeah, my suspicion is that what Aisha and what Matt and Abby were identifying here, because they were really focused on the anterior interpose, and in Aisha's case, it was a genetically identified subset of neurons in the anterior interpose, is that those neurons that you're most likely to find through perturbation or through recording, which Abby also did, are the ones that decelerate the limb. I think these are the same as one of the populations, the ones that go to thalamus and not the spinal cord. And in fact, if you look at, let me go back to the anatomy slide. The ones that go to thalamus and not the spinal cord are way more abundant, including an anterior interpose. So if you're just recording, you're way more likely to find these deceleration neurons. But if you can pull out the sort of needles in the haystack, these ones that are the yellow ones, the dual projectors, which are going to be very rare unless you go specifically, for example, into anterior or posterior interpose, or unless you're using this combinatorial viral approach for perturbation, I don't think you're going to see them. So we've sort of biased our recording sites and our viral targeting to these dual projectors because we think they're really interesting. And it turns out that they have the opposite function, this outward movement. And so I think what we're going to find is that in order to identify some of the discrete functions of these classes, you're really going to have to anatomically tease them apart rather than treat all of the neurons within a particular nucleus or subnucleus as homogeneous.
Wonderful. Yeah.
Okay, I'll ask another question because I really loved the work that you did on the nature of top-down and bottom-up sensory gating of brainstem neurons. And so the question you would think that the cerebellum would influence those top-down inhibitory or modulation of the inhibitory guys surrounding. So do you know whether the cerebellum influences those guys disynaptically or maybe trisynaptically?
Sorry, whether the cerebellum influences the
cortical neurons that you showed. You mentioned this early in your talk that provide the surround inhibition, or that's the way I encoded it anyhow.
Yeah, no, that's a good question. We haven't found, we did a comprehensive rabies tracing from those neurons, and we didn't find any significant cerebellar inputs, if I remember correctly, but I will double check after this. So I think I think that you're bringing up sort of an interesting, bigger question that I think somebody asked. Mark Churchill asked me this recently, and we sort of got into a discussion about it. So if you want to attenuate feedback, you can imagine that you'd want to do so with different levels of resolution. In some cases, you just want this to be sort of a blunting of feedback generally that's kind of messy and not very specific. So, and we see evidence for this. When a human is moving to reach for a target, you see attenuation of feedback, not just at the effector that's moving like the fingertip, but across the hand and even parts of the limb. So sometimes you just want to turn down reafference very broadly and throw out some of the good stuff with the reafference because it's just beneficial to do so. But sometimes you want really specific attenuation. This idea of like corollary discharge, like what Nate Sawtelle is finding in electric fish. And I think that's where the cerebellum really comes into play, because if you want to do very selective attenuation, both in timing and in modality, then what you want is a model to predict reafference and cancel it out. And so I think that what I showed in the cuneate is probably not well set up to do that fine grain attenuation, that corollary discharge type attenuation. I think it's just broad sort of turning down the volume. But where the cerebellum might come into play and where we haven't really studied its effects, where it might come into play with gain modulation, I think is in that predictive sort of negative copy of reafference.
Cool ideas, thanks.
right. Well, Iman, thank you so much for taking an hour, and thank you so much for voting today, everyone. Those who um, have the opportunity. I'm so pleased you had a chance to listen to your work. Thank you so much. Have a good day, everyone.
Thanks for having me.